Okay. Now, all right. The attendance sheet is there. The last time no one returned it. Make sure it goes all the way to the top and then return it to me.
Stop screen peeking.
I didn't know that was training.
Screen peeking? I know you use the one who won your live. You always screen peeking.
All right, so we are doing enzymes, and enzymes are proteins. But besides proteins, the enzymes also have a certain molecule or ion at the active site that is required for its activity. So when it requires such ion or molecule, that is called a cofactor. So cofactors are essential molecule or ion that is required for enzyme activity. Cofactors obviously can be of two types. It can be a metal ion, or it can be a small organic molecule. If it is a small organic molecule, that's called a coenzyme. That is needed for its activity. Now these metal ions or coenzyme can each be of two types. It can be loosely bound or tightly bound. So loosely bound, of course, means it goes in and out, and tightly bound means one metal ion is at the active site, never comes off. It's just 1-to-1 stoichiometric ratio. So the tightly bound ones you also need in less amount. The loosely bound ones you need in more amount because it is an equilibrium that goes back and forth. So the loosely bound ones are called the activator ions. And the tightly bound activator ions, because it activates the enzyme. And these are usually examples, maybe the group 1, like some of them may need potassium, ammonium. So group 2 will magnesium, calcium. So these metals usually are at the active site but not tightly bound. And as we saw before, magnesium is needed for anything that uses ATP, and magnesium helps to attract the negative charge of the phosphate. So the tightly bound ones are called prosthetic groups. The prosthetic group is a term used for anything that is tightly bound to any other molecule. It doesn't have to be an enzyme. For example, we already studied hemoglobin. In that, the heme is tightly bound to the hemoglobin. So heme is a prosthetic group of hemoglobin. So in the same way, if a metal ion is tightly bound to the enzyme, it is meant to increase activity. And these are usually the transition metal elements like zinc, iron, copper, so elements like that. So of the coenzymes, again, they can be of two types. These can be loosely bound or tightly bound. And if it is loosely bound, that is also called a co-substrate. If it is tightly bound, again, is the same name, prosthetic group.
Yeah.
So those that are tightly bound don't have to be covalently bound. They can be, some of them are covalently bound, some of them are just tightly bound because of very strong interactions with the enzyme, with all the different kinds of interactions that we have seen. So the basic idea is that the co-substrate comes out after the reaction, and then it undergoes further reaction. We will talk about that in a minute. The prosthetic group always stays bound to the enzyme. Now, all these groups that are increasing the activity of the enzyme actually participate in the reaction. For example, let's take an example of loosely bound ones. Here is this. The example will be NAD, ATP. So NAD is a coenzyme. ATP we already saw, it is adenosine triphosphate. NAD is nicotine adenine dinucleotide. Nicotine, then adenine, dinucleotide. NAD is an oxidizing agent, so it participates in oxidation reaction. Any reaction that uses some oxidation reaction will use NAD as the oxidizing agent. Another one is FAD, which is flavin adenine dinucleotide. So one example of a tightly bound is FAD, which is flavin adenine dinucleotide. And the difference between the co-substrate and the prosthetic group coenzyme is that after it is participating in the reaction, the NAD becomes NADH. Because it is an oxidizing agent, that's how it helps the reaction, so itself gets reduced, so it becomes NADH, that means it adds a hydrogen. And then the NADH must convert back to NAD for catalyzing the reaction again. So in order to help the reaction have activity, you need the NADH to be converted back to NAD. If it is a co-substrate like NAD, this NADH leaves the enzyme, then it goes into the cytosol where it is reacting or in the mitochondria. And then there will be another enzyme that converts it back to NAD. So because it is loosely bound, NADH converts back to NAD by a different enzyme, and then this again binds to the enzyme and gives you activity. It is not really waiting for this molecule of NAD to come back, but there is always a certain amount of NAD in the solution, so it always has some NAD to bind to. But the idea is that the product leaves and then it is reconverted back to the same one by a different enzyme. In the same way as we saw for ATP that participates in the reaction, it is converted to ADP, and the ADP then leaves. And then some other enzyme will convert ADP back to ATP. Yes?
So you said the product leaves and is reconverted back
to what it was before by a different enzyme. So it has nothing to do with this enzyme. Some other enzyme will do that. Whereas the tightly bound ones, like FAD, is also an oxidizing agent. And it is reduced to FADH2. And this cannot leave the enzyme. That's what the structure of the molecule is. It is tightly bound to the enzyme, it stays there, and so it has to get converted back to FAD by the same enzyme. It is not leaving the enzyme, and only when it is converted back to FAD then it can participate in another round of reaction. All right, so these are the classification which before we go into examples of any of these metal ions or other ones. There is another classification of the coenzymes. That applies to higher organisms like mammals, and that is dependent on the source of the coenzyme. Where do you get this coenzyme from? Do we make it or do we get it from outside? So those that we cannot synthesize, the higher organisms cannot synthesize many things. The lower organisms synthesize more things, like bacteria are capable of synthesizing almost anything that they need. They don't need anything from outside, other than the nutrients. So for this classification applies only to mammals, that we have to get some of these in our diet, and those are called vitamins, right? So everyone knows about vitamins that we need in small amounts, organic molecules that we need in small amounts, and we cannot make them. So they are vitamin-derived enzymes, vitamin-derived coenzymes. When vitamin was initially named, it used to be spelled with an E at the end. It was vitamin ending with an E, because people thought that these are essential amines. But then they found that none of these are actually amines, so they dropped that last E. So these are just organic molecules that are needed for enzyme activity, and they are actually converted to coenzymes, and they are part of enzymes. And then there are some that we can make, and so they are called metabolite coenzymes, which means they can be made in the body. So for example, as you all know, we make our ATP. So ATP will be one example of metabolite. Is a term used for all the intermediates in all chemical reactions in the body. There are numerous chemical reactions, hundreds and hundreds of them, and any reactant or product, they are all termed as metabolite. So example will be ATP because we make them. Example of vitamin, you know all the vitamins, there's a list of all the vitamins in your book. I also send you a list and what coenzyme they form. NAD is one of them. NAD, of course, is not itself a vitamin, it is derived from a vitamin. Always remember that these structures, the NAD, ATP, these are molecules that don't even cross the membrane. They have so much of charge on them. So they cannot be also vitamins that you can get in your food. So NAD is derived from niacin, for example. That is a vitamin that is then converted to NAD. So there are reactions that we do to alter the vitamin to make the final coenzyme. And then there are many others like vitamin B6 called pyridoxal phosphate and thiamine pyrophosphate. Now, each of these vitamin-derived enzymes, or whether it is a metabolite coenzyme, talking about the reactions that they catalyze or help catalyze, is of no use now because we haven't come to those reactions. So this chapter on coenzymes we will revisit every time we go talk about a certain metabolic pathway. For example, thiamine is a vitamin that forms thiamine pyrophosphate, which is a coenzyme. What it does, no point in talking about it now. We will talk about that when we discuss pyruvate or the citric acid cycle, because that's where it is used. So then we will again come back to this chapter on coenzyme and talk about that. For example, biotin is another coenzyme that we will talk about when we discuss how carbon dioxide is used to make organic molecules. Or in the same way, there will be many others, like coenzyme A is another one that we will need for the fatty acid synthesis. So we will talk about those at that time. So for the metal lines, going back to the metal lines, I already mentioned before that magnesium or anything that all metal lines obviously have a plus charge. So if it is at the active site of an enzyme, most usually all the ATP utilizing enzymes have a magnesium, and the purpose of that is to bring the ATP to the active site so that it can bind. So ATP is adenine, then ribose, and then 3 phosphates. As I said, the P with the sulfur base, each P has 4 oxygens, and so there are negative charges, and the magnesium helps to bind to the negative charges. So that's how it increases the efficiency of binding. Yes?
Is this, we're out of coenzymes, right? This is back to metal ion cofactors?
Yes, these are metal ion cofactors. We will very soon get into the coenzyme also. So one example of a prosthetic group metal ion is zinc.
What was just on the board just then?
Magnesium for anything that uses ATP as a substrate.
Magnesium is used for anything.
Any enzyme that uses ATP, because magnesium at the active site attracts the negative charge of ATP, and that's how it helps it to bind. Zinc is a metal ion that is present in the enzyme carbonic anhydrase. So carbonic anhydrase catalyzes the reaction of water and carbon dioxide. So it forms bicarbonate. And carbon dioxide has a low solubility in water, so this is a very slow reaction. That's why you need the enzyme to speed it up, and this enzyme is a very fast enzyme. If you don't have an enzyme, if you're doing this reaction in the lab, you know that carbon dioxide is an acid, so this will react with water much faster if you have sodium hydroxide in there. So instead of water, if you have a base, hydroxide, that's a strong base, that will very quickly react with carbon dioxide to give you bicarbonate. Right, so the carbon dioxide, I mean, I'm not going into the mechanism, but basically this is like carbon dioxide, this carbon with double bonds to oxygen, the negative charge, the hydroxide being a very strong nucleophile reacts at this carbon, and so that's how you get bicarbonate ion. But of course, in the cell, you cannot use hydroxide, everything's at the pH 7. And that's where the enzyme comes in. The function of the zinc in the enzyme is to create hydroxide ion at the active site. Even though you are in water at neutral pH, inside the enzyme, inside any protein is not the same as the solution outside. Inside it can be any strong base made, and this is how the zinc actually makes that strong base. So zinc, so here is your enzyme. And there is a zinc ion at the active site somewhere. And of course, for the zinc, remember zinc is the prosthetic group, so it must be very tightly bound. So it must be binding to something at the enzyme, and they all found this. There are histidines here. Histidine. So histidines are at the active site that interact with the zinc and keep it there permanently. So it's a prosthetic group. And the function of the zinc here is to, and then there is some basic group here. As I mentioned, this is how all enzymes function. They have the right base at the right time, at the right position to react with the substrate. And the base can be for example, a carboxylate ion that will remove a proton. So there is a certain base here, basic amino acid, that will remove a proton from water. So this removes the proton and these electrons go on the oxygen and the oxygen forms a bond with the zinc. So here you have the zinc, the base became protonated from this hydrogen and the zinc is now attached to the hydroxide ion. Yes,
Can you make that a little bit bigger?
you want a bigger active site.
Yeah, please.
And look at this guy.
All right, so here is the active site with the zinc, and that is bound to histidines here. And there is a basic amino acid here, and once again, bringing that back again, the basic amino acid in the protein is the opposite of basic amino acid in solution, right? Aspartate is an acidic amino acid. In the protein, aspartate is a basic amino acid because in the protein it is COO minus. Or same way lysine, arginine, these are acidic amino acids. So when I write a basic amino acid, it's most likely a aspartate or a glutamate there. So that will remove a proton from water. And then these electrons are used to form a bond or a partial bond to the zinc. Alright, so this bond to the zinc is not a permanent bond, so that means the hydroxide has some negative charge on it. It is just attracted to the zinc or has a partial bond to the zinc. So you are forming a hydroxide ion, even though not with a 100% full one positive charge, but significant charge on the hydroxide ion at the active site, which is impossible to think about having hydroxide at neutral pH. But the enzyme does that, it creates that hydroxide ion, and then it participates in this reaction. So the rest of the step is the same as here. So you have carbon dioxide, so the hydroxide reacts here, it's a nucleophilic reaction, and that gives you bicarbonate, and then that leaves. And then another water molecule comes in. So that's how the zinc participates in the catalytic mechanism. And remember, this is a very fast enzyme. This is happening at the rate of 10 million per second. So extremely fast, the molecule carbon dioxide comes in, water comes in, forms hydroxide, and then reacts with the carbon dioxide. So that's how, these are just examples. There are many other examples of metal ion activated enzymes. We are not going to discuss all of them. Let's go to the coenzymes. Oh, what have we? Yes. You want that written again?
Seems a little red.
Yes.
So you said something, you said a basic amino acid in the protein is not a basic amino acid in.
Yeah, we talked about it so many times. So if you have a free amino acid, like aspartate, that's an acidic amino acid. It's called an acidic amino acid. I didn't name it, but it's classified as an acidic amino acid because it has an extra carboxyl group. But at neutral pH, that extra carboxyl group is always deprotonated because all carboxyl groups have pKa about 4.8, that is much below 7. So no matter what the books call it, they will call it an acidic amino acid, but in solution at neutral pH it is always a carboxylate ion, and it can only act as a base, not as an acid. And in the protein also, because we are all at pH 7, so that means they are all acting as bases. Alright, so anything that is listed as an acidic amino acid never actually behaves as an acidic amino acid, even in solution in a test tube. Because if your solution is at pH 7, they are all carboxylate ion. Only time it will behave as an acid if you have a very low pH. Same with the lysine and arginine, they are not really bases. They are classified as bases. by the book or by the author or whoever wrote it, but it's never really a waste. It is always present as an asset.
This is my rewatch. We shouldn't.
All right, so as I said, most of the coenzymes we will discuss them when we come to those. But a couple of points to talk about here, for example, NAD and NADH. If you are doing such a reaction in the lab, let's say you are doing an experiment, and you want to measure the enzyme activity, you can measure the enzyme activity by either measuring the reactant, the substrate, and see that it is decreasing, or you measure the product and show that it is increasing. So you have to measure the rate. All the kinetics that we have done is the rate of formation of product. So you can measure the product. Another way of measuring the rate of reaction is to measure the amount of coenzyme. So whichever is easier to measure, that is the technique you will use to measure the activity, and that is called an assay. Every enzyme must have an assay, which is a method for detecting its activity. So one method that you can use for NAD containing enzyme is to measure the amount of NADH, because NADH has a very clear absorption at 340 nanometers. So if you look at the
Professor, sorry, what does assay mean?
assay means a way to measure activity.
Measure.
So if you're doing any work with any enzyme, you must have an assay. All the things that we talked about, enzyme kinetics, they are all about rates of reaction. How do you know what is the rate? You have to do that experiment, and that is done if you have an assay for that enzyme activity. Which can be, if the enzyme uses a coenzyme, you may be actually detecting the coenzyme, which if it is easier, which in this case it is, because the easiest assay for anything is to take your reaction and put it in a spectrophotometer, and it gives a reaction, it gives an absorption. That is the fastest assay you can do for anything. So you are always looking for a difference in its absorption spectrum, either the reactant or the product. If you find one, then you have a quick and easy assay. So any enzyme that uses this is usually assayed like this by looking at the NADH, because if you have NAD, it will have absorption like this, where this is around 280 or so. I may not have the right number, but somewhere in that range. And then this is NAD. And if you have NADH, it will have a new peak at 340. So NADH has a peak at 340 nanometers. So if you set your spectrophotometer at 340 nanometers, you can monitor the rate at which the product is formed.
Professor,
Yes.
would you consider the NAD as, I'm so sorry.
Yes.
As the NAD as the first one and then the NADH as the 340?
The first one is NAD, which absorbs here. NADH also absorbs here, but at this wavelength only NADH absorbs. So this is NADH.
You asked the same question.
And this one, NAD does not absorb there. So if you measure the absorption at 340, that's how it is always done. And if you see absorption, that is proportional to the rate of reaction. So that's how you do enzyme kinetics, by measuring the rates. All right, so as I said, there are lots of other things to study in coenzyme, but we won't do that now. We will do those as we go through the different pathways, and when we come to that enzyme, using a special coenzyme, we will talk about those reactions. So the next thing that we want to talk about are the metabolic pathways. In fact, the rest of the semester is all about metabolic pathways.
so much tighter. You don't have to turn your CT.
So pathways means how your different metabolites are utilized or synthesized. You can have how glucose is broken down, or how glucose is made, or how starch is made, glycogen is made, fatty acids are made, or fatty acids are broken down. There are all metabolic pathways. So the rest of the semester is on these various pathways. So metabolism is a very misunderstood term. The common public understands only weight loss and weight gain from metabolism. But that's not what it is. Metabolism means all the reactions that happen in a living cell. They all come under metabolism. Alright, so there are then numerous reactions. They are all under metabolism. And so every reaction, every product, every intermediate that is formed in these reactions are called metabolites. And because we have different types of molecules that you do these reactions with, and usually you can classify them as the different food groups like carbohydrates, lipids, proteins, nucleotides. So studying these will be called the branches or segments. So different branches or segments in metabolism, with the examples being carbohydrate, lipids, proteins, nucleotides, and so on. So these are the different branches or segments of metabolism. Within each branch or segment, there will be several series of reactions, each one called a metabolic pathways. So metabolic pathway is a series of reactions. For example, you may have heard of glycolysis that breaks down glucose to pyruvate. Or you can have gluconeogenesis, which means making new glucose, gluconeogenesis, which means you convert pyruvate to glucose. So there are a series of reactions that do that, and all that series of reaction is called a pathway. So this is what we will be studying. The metabolic pathways means the series of reactions starting from something and ending with something. And they are given names like glycolysis and so on. So the first question that comes to mind is, where does a metabolic pathway start and where does it end? Right? If we are talking of glycolysis, where does it start and where does it end? And the answer is, metabolic pathways don't have a beginning and don't have an end. It's all for our convenience and our understanding that we have named these pathways and say glycolysis starts from glucose and ends with pyruvate. Your cell doesn't know that today you are studying glycolysis and so you have to start from glucose and end with pyruvate. The cell has reactions before glucose and cell has reactions after the pyruvate. So for the cell, it doesn't make any difference whether you are studying glycolysis or citric acid cycle or any other cycle. It is our description of these pathways so that students have to study from here to here for this, and the next chapter will be on another set of enzymes starting from here to here. Alright, so in reality there is no beginning or end of a pathway except for anything that is coming from outside or going to the outside. There you have a beginning and an end, like food coming from the digestive system or anything that is being excreted, that's the end of a pathway, or starting with the food is the beginning of a pathway. Otherwise, there are so many other pathways with names, they don't really start anywhere or end anywhere, it's just that the books describe it as starting and ending. Do you have a question?
Yeah, I was just gonna ask, like, what does that say next to protein?
Protein, nucleic acids. which means DNA and RNA. All right, so metabolic pathways can be of three types. They can be linear, they can be cyclic, or they can be spiral. So the linear pathway, as the name suggests, you start from some reactant, then it converts to B, then it converts to B, converts to C, C converts to D, and D converts to E. And where do you stop? You stop where the book says you stop. Or for no reason, that's where it stops. But for the cell, it doesn't stop, it just continues from there. We have defined a pathway from here to here. So anything that goes in this fashion is a linear pathway. For example, you can go from glucose to pyruvate. That is a linear pathway. You can have a cyclic pathway. As the name suggests, you start with something and you end with the same thing. So you will start with A, and then A converts to B. So if you are starting with a certain compound, then at the end you get back the same compound, and that is called a cyclic pathway. And whenever you have that, the obvious question that comes to mind is, what do you achieve? If you start and end with the same thing, what is achieved in this? So what is achieved is that somewhere in this pathway, another molecule will enter. So A doesn't just convert to B, A combines with X, and then you get these different intermediates. And then at various points, usually smaller molecules than X will come out. So let's say X is a 2-carbon compound. Then 2 carbons will come out as carbon dioxide. So the purpose of this is to convert the 2 carbons of the starting material here to carbon dioxide molecule, and in the process you are using these intermediates that keep making them again and again. And that's why it is a cyclic pathway, and you are using the same enzymes again and again with every cycle. The net result is that you are converting the 2 carbons of this compound to 2 carbon dioxide molecules. Yes.
For linear reactions, you are also using the same enzymes over and over, right?
For another set of reactions, yes.
Like not just for cyclic, but for linear as well. You're also reusing the same enzymes, right?
Enzymes are always reused,
Yeah.
but you are not reusing with the last one as your substrate. You are always reusing with, let's say, these are the names of the enzyme or substrate. So if you have this, if these are the names of the substrate, then this is E1, E2, E3, like that. So if A goes to B and then to C and then to D, E1, E2, like that. So E1 will always use A as the substrate, and it will be a fresh A. A is not regenerated after the end of the reaction. So A is all converted to D and then to other products. You don't get back A from these. A has to come from somewhere, either from your diet or from something stored there. A has to come from somewhere. But in a cyclic pathway, the A is not coming from anywhere. is always there in a certain amount, and that is the one amount that is getting regenerated again.
Thank you.
And so, based on your question, another thing you can expect is that the concentration of A usually will be very low because you are regenerating it again and again. So in this case, the amount of A depends on where it is coming from. For example, if it is glucose, then glucose can be very high concentration, so A can be very high. But in a cyclic pathway, all you need is small amount equal to or similar to the amount of enzyme. You don't need more than that, so because it will again be regenerated. And so all the intermediates in a cyclic pathway are present in small amount and they are regenerated, and that is the definition of a catalyst. So that means in a cyclic pathway, each intermediate is also considered as a catalyst. In fact, this is how the cyclic pathways were discovered. Citric acid cycle, which is also known as what? The Krebs cycle, and Krebs discovered that by discovering these intermediates as catalysts. So he took mussels and found that the rate of reaction increases if you increase, for example, the amount of citrate or succinate. So they are present in small amount. If you increase their amount, the mussels have more activity. And that's how the citric acid cycle was discovered, by showing that each intermediate is a catalyst. In a spiral pathway, so in a citric acid cycle, this is acetate. We are going to discuss that in more detail later. And the purpose of the citric acid cycle is to convert the 2 carbons of acetate to 2 carbon dioxide molecules. In a spiral pathway, you start with the compound A, and then it will go through a series of reactions and convert A plus, let's say, 2 carbons. So you get a new compound which is longer than A by 2 carbons or 3 carbons or whatever. So it's adding carbons to A. But the functional group is still the same. So this will again react in the same way using the same enzymes and then convert to A plus 4 carbons. So in every cycle you are increasing the chain length by 2 carbons, as an example. And this example comes from fatty acid synthesis. Fatty acid synthesis happens this way. You are using the same enzymes. So if this is E1, you are again using E1. If this is E2, you are again using E2. So you are using the same enzymes, but in every cycle you are increasing the chain length by 2 carbons. So it is not the same substrate, but similar substrate, differing by a certain number of carbons. And that's why it is called a spiral pathway. You keep doing this in fatty acid synthesis. You keep doing this till you have made a 16 carbon fatty acid called palmitic acid. The same will apply for degradation. If A has 16 carbons, in degradation you go through a series of reactions and then you get 14 carbons. And then use the same enzymes again and do another round of reaction that gives you 12 carbons. So in degradation also, you are removing 2 carbons at a time in each cycle. So when that happens, that is called a spiral pathway. And as I said, we will discuss these pathways in more detail: fatty acid synthesis and degradation, or glucose converting to pyruvate, or using the citric acid cycle. Yes, sir.
Is it always carbon?
No.
If it is doing that type of reaction, it doesn't have to be. The idea is that it is using the same enzymes to create a similar molecule, but not the same molecule. All right, so we will stop here now. Next class, we will look at anyone who has
Bro, please stop on the heart, please. Remind me. Yo, watch your mouth, bro. Yo, all I do, bro. Watch your mouth, bro.